Hello students, you made it to the first day of the last week before Thanksgiving. Thanksgiving's a great holiday, probably one of my favorites because you get to eat so much. Whether it be a rosemary stuffed turkey, whole carrots, lots of potatoes, and then let's not forget about the best part, dessert. AKA, you're gonna be consuming a lot of sugar, AKA glucose. In today's lesson, we're going to figure out what happens to that food once it actually goes through your digestive system and gets to your cells. AKA, how do you turn those biomolecules, mainly glucose, into usable energy? ATP. Let's do this. The process in which glucose, or any biomolecule for this matter, gets broken down is a catabolic reaction because you're taking a big biomolecule and breaking it down. While doing that, you're also going to release a bunch of energy in the form of ATP. And this is the energy molecule that our bodies can actually use, the one that breaks off its third phosphate to release energy. This process is called cellular respiration. Cellular, it relates to cells. Respiration, because there's an exchange of gases that occurs. It's also sometimes called aerobic respiration, like the air that we breathe. By now in class, you have learned that this occurs in a little bean-shaped organelle called the mitochondria. Let's see what this mighty organelle is all about. So we'll start with this diagram that if you're able to, you can print off of the hub, and if you're in person, you should have already. If you're not able to, go in and find yourself a little blank sheet of paper. We're gonna make another cool foldable. If you're using a blank sheet of paper, you're gonna wanna fold it in half. Sorry, I do have something printed on the other side of mine, but let's pretend it's blank. Like this, and you're going to focus like we did for the photosynthesis one on the bottom half of the diagram. Now, if you're at home thinking, Miss Lim, how on earth am I going to make this diagram on my own? Do not fear. First of all, use a pencil so that you're able to go back if needed. Let me show you guys the basic layout of how it needs to happen. If you've got the template, you can skip forward a little bit. So you're going to need to start with glucose, which is a hexagon. You're going to have an arrow pointing to a box of some kind. And then this box is going to go down into a mitochondria. Now, a mitochondria looks very much like a little bean, so I would just draw a rough bean shape for the rest of my page. Okay? Mitochondria have two membranes. They've got an outer membrane and then they have an inner membrane. The inner membrane is kind of weird. He looks like very squiggly, kind of like this. So you're going to want to make your inner membrane with all these folds called cristae. Okay? From here on, I think you can follow my diagram well to see what you need. So like as I talk, you can draw arrows off of the main arrow. You can draw a circle as needed, etc., etc. And I think you'll be okay. Cellular respiration occurs in 3 parts. The first part is called glycolysis, which literally means sugar splitting. Let's see why. Like I said, cellular respiration happens in 3 steps. The first step is called glycolysis. Now, if I look at this word glycolysis, what I'm seeing basically is sugar. And lysis, remember, is to break apart things, a sugar break. And that is exactly what is going to happen here. So we start off with glucose, our favorite biomolecule, giving us lots of short-term energy, C6H12O6. And through step 1 of glycolysis, I'm going to break glucose apart into 2 molecules of something called pyruvate. Pyruvate has 3 carbons, glucose has 6. So we can imagine in some ways glucose being literally split in half so that I form 1 molecule of pyruvate and a second molecule of pyruvate. Let's record that up here. So we've got glucose being split in half into 2 molecules of pyruvate. This occurs through the action of enzymes. in the cytosol. So we're not even inside the mitochondria yet. As you can tell in my diagram, we are outside in the cytosol. Now, of course, to kickstart this reaction, because glucose is a fairly stable molecule, I actually have to add in some energy. That will be in the form of 2 ATP molecules. So 2 ATP molecules come in, they break apart their phosphate bonds. That gives enough energy to break these bonds between the glucose carbons and give us our pyruvate. So we've got an input of energy. 2 ATP are going to be used to power the lysis. Now, of course, as soon as I break the bonds, I also release a fair amount of energy. So interestingly enough, 2 ATP net come out. In reality, 4 molecules of ATP are created in the process of glycolysis, but if 4 are created and 2 are used, that gives me a net of 2 ATP in this process. Already starting to make ATP, awesome. When I'm breaking the bonds, I'm also going to release some electrons. And as you can imagine, electrons always like to find their electron transporter. So we have some electrons here released, and they're going to go on to a good old friend, NADH. You might notice it's not NADPH, and that is because we're working with cellular respiration, but it has the same concept. NAD plus will become NADH. We can record that here. So there will be 2 free electrons created, 1 for each molecule of pyruvate, and they're going to go onto the electron transporter NADH. And that is it for glycolysis. I am inserting 2 ATP to break apart glucose. I'm going to glycolyse, or split my sugar in half, into 2 molecules of pyruvate. During that splitting, I release enough energy to create 2 ATP and to collect some electrons. We're going to move on to step number 2, which, as you can see, is a cycle. This one has multiple names. We'll put in a few. So the most well-known one is the Krebs cycle. It is also sometimes called the citric acid cycle. And that is because, much like the Calvin cycle in photosynthesis, citric acid is used. I'm going to stop talking so I can actually write. Citric acid is used to bind pyruvate as it enters. Now, along the way from going from the cytosol into the mitochondria for the citric acid cycle, pyruvate does get transformed one more time. He's transformed into a molecule called acetyl-CoA. And when he does this, he will release a little bit of carbon dioxide waste. So we've got pyruvate. Pyruvate is going to be transformed into acetyl-CoA. And this is going to release some carbon dioxide. Basically what I'm doing is using a bunch of enzymes to break down the 6-carbon glucose into a 3-carbon pyruvate and now a 2-carbon acetyl-CoA. I'm breaking down my big molecule. Inside the mitochondria, that is called the matrix of the mitochondria, very much like the stroma in the chloroplast. It's just a jelly-filled area. And this is where the citric acid cycle is going to occur. Matrix has its own special proteins or enzymes that are going to help break down acetyl-CoA even further. He's basically going to break down acetyl-CoA until it is nothing but carbon dioxide gas, our waste product, only 1 carbon. So it went from 6 to 3 to 2 to finally just 1. During this process, as I'm splitting apart acetyl-CoA, I will release some ATP. Only a few molecules, 2 ATP, will be created. And then finally, the most important part of the citric acid cycle, I'm going to harvest a whole bunch of electrons. I'll have some electrons that hop onto NAD+, the electron transporter, and make NADH. Now I'll also have 2 electrons at a time that can hop onto another electron transporter called FAD+. He takes 2 at a time and he becomes FAD H2. Lots of electrons being created. And that is it for step 2. Let's fill in some blanks. Acetyl-CoA is going to enter the mitochondrial matrix, the very inner part of the mitochondria. The enzymes are going to break it down further into many, many things. We've got CO2, which is going to leave as waste. We've got 2 ATP. And we've got many electrons. The name for this cycle, if you want to test yourself, there is 2 names. It can be called the citric acid cycle or the Krebs cycle. Finally, what happens to those electrons? The electrons are always harvested by electron transporters. We've got 2 types here in the citric acid cycle. We've got NADH, and then we've got the double whammy one who picks up 2 electrons, FADH2. Moving on to step number 3. So as you can see so far, if we follow the path of our carbon, we broke down from 6 to 3 to 2 to 1, and he's basically just waste. That's it. We were just breaking him down. We've collected some ATP, and we've been collecting a bunch of electrons. Step number 3 is where the electrons get to shine. This is the electron transport chain. And it's also known as the ATP synthesis or chemiosmosis portion. So let's follow these electrons. If you want to, you can color in this arrow. You don't have to. I just like following them here. Where these electrons go is to something that looks exactly like the one in photosynthesis. It's a bunch of proteins along the membrane. If we were to label our mitochondria a little further here, we would notice that there are 2 membranes. There's this really squiggly one with a bunch of folds. This is going to be your inner membrane. And then, of course, we have the outer one, the one on the outside. This is our outer membrane. And the cool thing that this does for us is that it gives us an intramembrane space, a space between the membranes where we can play around. Yeah. Back to the electron transport chain. Electron transport chain is a name for these proteins that hang out here. And as the name implies, we can put some more around here if we want. They're all over this membrane. Electrons that were collected earlier are going to hop off of their transporters and head on to these electron transport proteins. Now they will travel from protein to protein to protein to protein to protein all along the membrane. As they are traveling, they're binding to each protein, and they're causing it to change shape, pumping a hydrogen ion across into the intermembrane space. This is going to happen for most of these proteins. So we can see that we start to build up a really high concentration of hydrogen ions in the intramembrane space, the space between the 2 membranes. Let's capture some of these ideas in fill in the blanks. So our 2 electron transporters, we have NADH, who's carrying 1 electron, and we've got FADH2, who's carrying 2. They're going to carry their electrons from glycolysis, the first step. They're also going to carry electrons from the citric acid cycle, the second step, to the inner mitochondrial membrane, where the electron transport chain is occurring. Okay, then the electrons are going to jump off of their electron transporter, and they're going to go to a bunch of proteins in a row on the membrane, better known as the electron transport chain. As they travel along each protein, they cause it to change shape and pump a hydrogen ion into the intermembrane space, creating a nice high concentration inside, low concentration in the matrix. So we're creating a hydrogen ion concentration gradient. You can probably see where the setup is going. Now, just like in the chloroplast, we have our friend, the ATP synthase, a very special protein channel that allows facilitated diffusion of these hydrogen ions back through into the mitochondrial space. So the hydrogen ions can flow back through this guy to their low concentration where they want to go. And as they do that, this protein is actually going to be turning around on itself, very much like a crank on a turbine or a crank on a windmill, water mill, any of those sort of things, and creating kinetic energy. And as he's cranking and creating his kinetic energy, he is actually going to be spitting out ATP molecules. So this little guy, the ATP synthase, and y'all can definitely draw yours a little bit nicer than mine if you would like. He's going to turn and crank himself around, kind of like that. And he's going to allow H+ ions to travel through him from high concentration to low concentration. And as he does that, he will force an ADP to get together with a phosphorus and to produce ATP. Now this, my friend, this right here is the money maker. This is the step that makes the most ATP. In fact, the number of hydrogen ions that you will have created a concentration gradient with and that flow back through will power the creation of 32 to 34 ATP. Wows! So what we're seeing is the hydrogen ions are flowing back into the mitochondria matrix through a protein called ATP synthase. And ATP synthase can create for me 32 to 34 ATP molecules. Crazy. This stage produces the most ATP, right? So compare it to glycolysis, only 2, citric acid, only 2. This one creates the most ATP by far. All right, we've got one last little molecule that needs to come in. And if I'm trying to remember my equation, it would look something like this. I've got glucose, C6H12O6, plus something, and that's going to give me 6 carbon dioxide, which I'm already seeing produced in the citric acid cycle. And then it's going to give me something. Basically, I'm missing the 2 biggest parts of my equation here. And those 2 biggest parts are 6 oxygens and 6 waters. And that is where these 2 molecules come in, only at the very, very end. Oxygen will enter the mitochondria, and his role is after the electrons are done bouncing from protein to protein to protein to protein to protein, the electrons are actually going to combine with oxygen. Not sure the best way to draw this, actually. And together, when I combine an electron with my oxygen and an H+ ion, I'm going to create the waste product of water. Interestingly enough, while oxygen and water are both very vital for us, in both photosynthesis and cellular respiration, they're really just there as accessory waste products. Let's capture this last step for ourselves. So the free electrons bind to oxygen at the end of the electron transport chain, and they're going to create water as a waste product. I'm actually going to make my oxygen blue because I kind of like having everything a bit color-coded. So here we have cellular respiration step by step. Step 1, glycolysis. Break apart glucose into pyruvate. Produce 2 ATP, make some electrons. Step 2, citric acid cycle. Move into the matrix. Use some enzymes to break apart acetyl-CoA into carbon dioxide waste, 2 ATP, and some electrons. Step 3, the real moneymaker. Take the electrons from the first 2 steps, use them on the electron transport chain along the mitochondrial membrane to create a hydrogen ion gradient. Let the hydrogen ions flow back naturally through facilitated diffusion around the ATP synthase. Let the ATP synthase rotate and crank out our ATP. Create 32 to 34 ATP. Woo! And then finally catch your electrons at the end of the electron transport chain using an oxygen molecule to create water. Ta-da! I realize I forgot to put in the steps here. Let's do this as well. So step 2 is called the Krebs cycle or citric acid cycle. Step 3 is called electron transport because electrons are doing most of the work and going from molecule to molecule. It is also sometimes called ATP synthesis, meaning I'm making ATP. And there you have it. You have the detailed steps 1, 2, and 3, glycolysis, citric acid cycle, electron transport for making ATP. Go ahead and take your foldable and fold him in half. Hopefully you did not use Sharpie like me so it bled through. And let's go grab a pair of scissors. Be careful, don't go all the way to the middle if you can help it. Something like this. All right, moment of truth. This is step number 1, or glycolysis. This is step number 2, or the citric acid cycle. Here is step number 3, or the electron transport chain. Let's go ahead and label the front of our foldable. Once again, sorry that mine bled through quite so much. So we've got glycolysis. How many ATP did we make? Only 2. Second step, citric acid cycle / Krebs cycle. And that's also 2 ATP. Final step, electron transport. / ATP synthesis. And that's going to be the big whopper, 32 to 34 ATP. Altogether, we make 2 plus 2, 4, plus 32, 36 to 34 ATP altogether. And remember, that would be cellular respiration, also sometimes called aerobic, as in the air I breathe, aerobic respiration. Nice work, students. You're going to want to glue this onto page 34. Glue it so that you're still able to lift your flaps and see what's underneath each one. Now, if you're thinking, Ms. Lim, seriously, this is so complex. What? Just think about the fact that it is happening in all of your cells at this moment. They're producing trillions of ATP molecules just so you can actually process this video and watch it. I'm always blown away by how amazingly complicated and how well functioning our bodies are. Pretty cool. That's it. I hope that you continue cellular respiration in all of your cells for the rest of the day. Do not forget your notes on the hub, and I will see you guys in class.